Welcome to session 4, which is the industry panel. And the industry panel is the opportunity for companies active in the Ataxia field to present their respective R&D programs. So we invited companies to this panel, and it's explicitly not in the sponsorship packages. So the companies we have invited, we really see as partners on our way to clinical trials. The presentations cover preclinical topics, biomarker topics, and trial results. And the companies which are going to present are Triplet, UniQure, Vico, IXICO, Cadent, and Biohaven. Each presentation will take about 8 minutes, and we have 2 minutes time for discussion. So we're running a bit late, and initially we thought we have 20 minutes for discussion after the presentations for a general discussion on trial readiness. We see how much time is left over. For now, I think it might be.
Thank you. I'll do my best. Let me start my timer. We're not as punctual as the Germans, the Dutch, but I'll do my best to stick to 8 minutes. So I'm happy on behalf of UniQure to present our gene therapy approach for SCA3. So it's an AAV-based approach. We use AAV5, and the intended route of administration is the Cisterna magna, which is the CSF area between the cerebellum and brainstem. So after injection, we know the AAV distributes to these brain areas and is taken up both by neurons and glia cells. Then the AAV translocates to the nucleus, where the capsid is degraded and the DNA content is released. And so this DNA stays present in an episomal state, is constantly transcribed into a microRNA. This microRNA is processed into only a guide strand. That's one of the unique features of our platform, that we don't have a passenger strand. And so this helps with limiting off-target effects. And this microRNA specifically binds the Ataxin-3 mRNA, resulting in total lowering of Ataxin-3 in a non-allele specific way. So in this short talk, I'll briefly run through some preclinical research that we did, from lead selection in cell models, some 3 mouse models that we're able to use, and distribution data from non-human primate study. So here we're looking at the lead selection that was performed in HEK cells. And so for this, we use a system where we have the Ataxin-3 gene linked to a luciferase. So it results in a measurable light signal. And we use this because it's a very robust assay with low standard deviation, as you can see. And so in the HEK, we co-transfect this Ranilla reporter with a range of microRNAs, and you see the data below. So a total of 11 microRNAs were screened. And for each of them, you see the dose response. And in the end, it was decided that numbers 7, 9, and 11 were most potent. Those were then tested at protein level, and in the Western blot here, you can see the knockdown of Ataxin-3, which is around 75% in this system. So another important check that we always do is checking the processing. So this is an experiment in iPSC-derived neurons. And here we're using the lead candidate AAV. And this is based on small RNA sequencing. So now we're looking at, in neurons, the top 60 endogenously expressed microRNAs, and in orange is then our lead Ataxin-3 microRNA. And you see it's the 3rd most expressed, so quite high level, but it's within the physiological range. And it's important to check this because it's known if you have a very strong promoter driving really high expression, this can saturate the RNAi machinery, leading to toxicity in that way. So we always check our lead candidates that were within the normal range. Now, this is the first in vivo study. This was together with Jeanette Hubener from Tübingen. And as was shown yesterday, also they have this knock-in mouse model with, I believe, around 300 CAGs. And this was a big study, but we're showing now just a small subset of data. It was also investigated what was the most efficient route of administration, such as the ventricle or DCN. But in the end, Cisterna magna turned out to give best coverage of both cerebellum and brainstem. And so upon single injection in this mouse with our lead AAV, we see around 50% knockdown of Ataxin-3 in the cerebellum, and a little bit higher knockdown even in the brainstem. So this is a second in vivo experiment performed together with Luis Pereira from Coimbra University. And they have a lentiviral mouse model, and that expresses in striatum locally the human cDNA sequence for ataxin-3, so with expanded repeat, of course. And the nice thing about this model is that there's a strong molecular phenotype. And looking at the top panel here, this is a staining for ataxin-3. And you see in the striatum, there's a lot of aggregates or inclusions, as you want to call them, throughout the striatum. And here, 3 different doses of the lead candidate AAV were tested, so local injection in striatum. And with increasing dose, you see a reduction in the number of aggregates. And at mid and high dose in this experiment, the aggregates were completely removed even. And the second histology they performed was for DARP32, which is used as a neuronal integrity marker in this experiment. And you see that in the untreated mouse, there's a lesion where this marker is absent. And with increasing dose of our AAV, you see the lesion size reducing and going away in mid and high dose. So it's very promising preclinical data. And then the last in vivo, this was together with the University of Michigan. And there they have a transgenic SCA3 mouse model, the MGD84. Some of you may know it. And again, here we did Cisterna magna injection of 3 different increasing doses. And in this case, we're also looking at behavioral improvement. And on the left, you can see the vector DNA levels that we get in cerebellum and brainstem after the Cisterna magna injection. And we get a good coverage of cerebellum, and particularly brainstem is targeted with very high levels. Now looking at the behavioral, so this is total locomotor activity, so a home cage system where the mouse can just freely move around. And then in gray, you can see the vehicle-treated homozygous mice, and in black is the wild type. So you see at start of injection at 8 weeks, there's already a big difference. And over time, we do see some stabilization with all 3 doses, so the decline is not as big as an untreated. But maybe in this case, we're already a bit late with the treatment if you see this difference. So that's something we wish to investigate as well in the future. Now we move to the non-human primate experiment. And this was an experiment to check whether this Cisterna magna is also suitable in the larger brain. And here the main question was to check which volume do we need to inject, also to relate to the patient in the future. So in the end, we did 3 volumes up to 2 mLs. I'm just showing the 2 volumes here to keep it a bit more easy to interpret. So again, Cisterna magna injection with these 2 volumes. You can see some coverage in cortex, midbrain, but especially the areas of interest, so brainstem and cerebellum. We get similar levels as we know are efficacious in the mice. So that's good news. Oh, excuse me. And also interesting for SCA3, using the Cisterna magna, we get really good coverage of the spinal cord, which is good to see. And with the high volume of injection, you get better coverage of the lumbar regions. So again, from the non-human primates, here the question is, is the processing of the microRNA the same as we see in the mice and in human cells? And luckily the answer is yes. So you can see in the x-axis the copies of vector DNA, and on the y-axis the number of microRNA molecules, and you see a good correlation between those two. And orange, the high volume, you can see that you get higher levels of both vector DNA and microRNA expression. So this shows indeed the functional molecule is produced in this experiment. And at the moment we're working on in situ to image also more clearly. So how is the distribution in these primates? So on behalf of UniQure, I really have to thank all the collaborators, many of whom are in the audience here. We're really honored to work with some of the leading SCA3 researchers, as you can see. So the group of Henk Post and Haley McLaughlin from Michigan, Jeanette Hubener from Tübingen, Willeke from Leiden, and Louis Pereira and Rui from the Coimbra University. So thank you.
Okay, thank you very much. So we have time for one question. And I just take the question from the Q&A, which got the highest ranking. So what is the biodistribution into the cerebellum after AAV injection, and how much in the dentate and how much in the cortex?
Yeah, so that's a good question. Well, in mice, the coverage is really good, as you can imagine. So for that, you usually use the GFP constructs. Cortex is covered to a smaller extent. You saw also the data from the non-human primate. It's about a log lower or so.
Okay, thank you very much. Then we move to the next presentation. And we retry Irina and Antonijevic. Can you please unmute yourself and then, yeah, okay. So screen sharing works. Wonderful. Please go ahead.
Great. Well, good afternoon, good morning, everybody. Thank you very much for the SCA Global Organizing Team to give Triplet Therapeutics the opportunity to present our approach to treat different repeat expansion diseases, which include spinocerebellar ataxias and other ataxias, include also Huntington's disease. And we're developing molecules to target the DNA damage response pathway, the DDR pathway. And the goal is to modulate somatic instability, which has been recognized as a key driver of multiple repeat expansion diseases. So this is my disclaimer. Repeat expansion diseases, or REDs, represent a major unmet medical need. Many of them are rare monogenetic diseases. Many affect the central nervous system. And even though the diseases are different in their presentation and they affect different genes, there are common shared mechanisms between those diseases, and this includes a DNA repeat expansion that, when reaching a critical threshold, causes the disease. This repeat expansion can be in different parts of the gene. In Huntington's disease and in several of the spinocerebellar ataxias, is in the exon. It's in one exon, so it's a protein coding sequence, but in other diseases such as Friedreich's ataxia, it can be in the intron. But what we have also come to appreciate is that this repeat expansion that causes the disease is actually not static, is not fixed at birth, but continues to expand. And this is what we call somatic expansion. And this is what Triplet Therapeutics is targeting. Now, I will talk a lot about Huntington's disease because a lot of the science has really sort of generated some of this novel thinking about these repeat expansion diseases comes from HD. But we have now come to appreciate that what we have learned in HD really is applicable to other diseases, and in particular several of the ataxias. And this is why we think it's of interest to think of a much broader spectrum of diseases that we can target with the same molecule. This is our goal. So what we have learned in HD is that the gene dose of the inherited mutant gene is actually not so critical for disease severity, but it is rather the longer inherited allele. And what we know today is that the longer the inherited allele, the more prone this gene is to exhibit somatic expansion. And in the middle panel, you can see here an example of what somatic expansion looks like. Somatic expansion does not occur in every tissue of the body, but in certain tissues, it is much more pronounced. And so particularly in the brain, in the striatum, and in the cortex, we see typically a lot more somatic expansion than, for instance, in blood cells. And what you can see here in green is the healthy allele, so it's at the bottom of this gel. Then you can see here the inherited mutant allele at a thick band. And then each of these black dots represents cells in the striatum or the cortex where the mutant allele has expanded way beyond the inherited length. And in this particular example, patient 1, who died several years prior to predicted age of onset with HD, had up to 1,000 CAG repeats in some cells, whereas the inherited repeat was only 41. So this shows you that what we can measure in blood is not representative of what is actually going on in the brain. And this also shows that this somatic instability in those brain areas that are so key for driving the disease really occurs way before the full motor manifestation. And as I said, this somatic instability, this sort of mosaicism in different tissues is clearly seen in multiple REDs. Here's an example, SCA1, ALS, as well as DM1. Now, this somatic instability now helps us understand why two individuals who have the same inherited CAG number can actually have a widely different age of onset. And GWAS studies, or whole genome association studies, have identified the DNA damage response, the DDR pathway, as really a pathway that is enriched in this genetic modifier analysis that has been conducted by Jim Guzella, Marcy McDonald, and Yeonming Lee at the Harvard Medical School, MGH. They have really done a 9,000 patient HD patient study, and they have identified multiple genes in this DDR pathway that clearly contribute significantly to age of onset. And one of those candidate genes, MSH3 in this example, has been looked as a candidate gene in a smaller set of patients, two different groups of patients, patients with Huntington's disease and myotonic dystrophy type 1. And what has been seen is that a lower expressing, so not pathogenic, but just a common variant which is associated with lower expression of the gene protein, was associated with less somatic instability, a later age of onset, and in the case of HD, a slower disease progression. Now, this is very encouraging in terms of a drug development approach, knowing that we can lower certain genes, and the expectation is that lowering this gene would have the impact in the brain on somatic instability and therefore also on disease onset. So Triplet has then developed antisense oligonucleotides, ASOs, against different genes in this DDR pathway. And one of our tool molecules, here called TTX3281, was injected in different HD mouse models. In this case, here is shown the Q111 mouse model, and we have injected the ASO ICV. But you can see here that the target gene was reduced by 50% in the striatum and cortex. And then on the right-hand panel, you can see that this reduction of the target gene really led to a very marked effect on somatic instability. Somatic instability index is a way to calculate somatic instability, where you calculate how many cells show a much longer repeat compared to the inherited one. And you can see that with this level of reduction of the target gene, there was really no expansion in the cortex, and in the striatum, where expansion has already occurred when we started to intervene, there was a marked hold. Really, there was a halting of further somatic expansion at the time of intervention. We have then developed multiple molecules that actually bind to the human gene. We have looked at multiple of those molecules in non-human primates and monkeys. And what you can see here is our clinical candidate, the lead molecule. We have injected this in the monkeys into the CSF. And you can see here again a very pronounced reduction of the target gene, about 47% in the caudate and about 77% in the cortex. Now, this is very encouraging because all the mouse data have shown us that this level of reduction should hold somatic instability. We cannot measure this in the monkey because the monkey does not have a mutant gene that would show somatic instability. But as we now think of our clinical development plan, and I was very happy yesterday to hear from multiple of the presenters that really the pre-manifest disease stage is also very important in spinocerebellar ataxias and other ataxias. And this is what we have seen in Huntington's disease. First of all, we know somatic instability occurs during pre-manifest disease stage, but there are also clinical as well as biomarker changes that clearly occur during this period of the disease. And the FDA recognizes the importance of that, and as part of the HD CPAS consortium, there has been a development of a cognitive battery designed to measure cognitive decline in this pre-manifest period, and there has also been development of a patient-reported outcome that we heard yesterday also how important this is when we think of intervention studies. Now, in preparation of our clinical trial, we have started a natural history study, SHIELDHD, where we will be measuring the same clinical outcome that we then plan to measure in the phase 1 clinical trial in the extension of the study. We are collecting blood and CSF, and those biofluid collections are very important for biomarkers. CSF in particular will not be only used for NFL, but also for target engagement biomarkers. We can measure our target gene at the mRNA level in CSF exosomes, and we'll be looking at MRI. And as we then think of our first in human clinical trial that is planned to start in the second half of 2021, we will be doing first a dose escalation study where we will look at a safe and well-tolerated dose, and we will be utilizing this target engagement biomarker to confirm that we have adequate engagement. And then this dose level will be taken into a phase 1 extension study where we will be measuring the different clinical and biomarker outcomes to help us conclude on proof of concept. And the SHIELDHD trial will be important either as a run-in phase or placebo augmentation. We heard this morning from David Clifford, we try to also minimize the number of patients that will be exposed to placebo in the study. Now, SHIELDHD is on its way. It includes patients with a genetically confirmed diagnosis of HD and patients who can undergo MRI scans, blood draws, but also importantly lumbar punctures, and can also complete the cognitive battery. The study is designed as a 2-year study with 7 visits over those 2 years. We plan to enroll approximately 60 patients, and we have divided the patients in 3 cohorts in an early prodromal, late prodromal, and an early manifest patient population to have the greatest density of data to help us again plan the phase 1 clinical trial. The study is well on its way. I would really like to thank our investigators and the participants who have been enthusiastic enrolling into the study despite COVID-19. The study is currently being conducted in 5 countries at 9 sites. And I would like to thank you, the audience, for your attention, and I will be glad to take any questions.
Okay, thank you very much. Again, one quick question, this time from Mattis. So why didn't you use the natural history data which are already available for Huntington's disease and instead started a new one?
Yeah, so this is a question we get a lot. And the problem is that we wanted to use some very specific outcome measures, which are currently not being integrated in the natural history studies that already exist, such as the HD cognitive assessment battery, at this frequency that we wanted to have so that we can actually have the greatest information for our phase 1 clinical trial. And the other really constraint was the access to CSF samples. We have developed an assay to measure our target engagement biomarker, and it is very important that we have better knowledge about the variability across those different patient cohorts that I showed you, and also longitudinally, and the access to sufficient CSF to do this assay is just not available. And we have discussed this with CHDI and ultimately felt that we needed to have this data to really be optimally prepared for the phase 1 clinical trial. Okay, thank you very much. Thanks again. So we'll go ahead with the third presentation, which is given by Nicole Datson from Vico.
Good afternoon. Good morning, everybody. Can you see my slides?
Yes, we do.
Yeah, I'll try to put them on full screen in a minute. Yeah, can you see it? Yeah, good. Okay, so my name is Nicole Datson. I'm from Vico Therapeutics, and I would like to discuss today the program that Vico is developing in order to suppress mutant polyQ protein expression using a single CAG targeting antisense oligonucleotide. And this antisense oligonucleotide is being developed to as a therapy for polyglutamine or polyQ disease, a group of 9 neurodegenerative disorders depicted here that all have a mutation in common, that is the expansion of a CAG-repeat in a coding exon. And this CAG-repeat expansion gives rise to an elongated polyQ tract in the corresponding proteins, which gives rise to a toxic gain of function of the corresponding mutant protein, which then leads to the neurodegenerative disease that is so characteristic for these 9 disorders. And what Vico is doing is we're developing a single antisense oligo for this group of multiple polyQ diseases. The oligo is called VO659, and it is a 21-mer that has a sequence CUG7, which is perfectly complementary to 7 copies of a CAG repeat. The chemistry is 2-O-methyl phosphorothioate chemistry with methylated Cs. And here on the right-hand side, you can see how the oligo works. So this is an example of polyQ transcript. It could be any one of the 9 different genes for the polyQ disorders. And you see here an expanded CAG-repeat. The oligo binds to the expansion of the expanded CAG-repeat present in the transcript of the corresponding gene, and by doing so sterically hinders translation, giving rise to reduced levels of the mutant polyQ protein. And on this slide, you can see again the 9 different disorders and the normal repeat length and the pathogenic repeat length. And what, of course, is striking is that each of these different disorders have their own threshold above which the CAG repeat expansion becomes pathogenic. And that is of interest with respect to VO659, since longer CAG repeat expansions present in the mutant alleles can harbor more copies of the oligo and hence give rise to a stronger reduction of the mutant protein versus the wild type protein that has the shorter repeat. And this is a phenomenon we call allele selectivity. And that is explained a little bit, or illustrated, I should say, on the next slide. So here you can see some in vitro data we obtained on a panel of different Huntington's disease patient fibroblasts. These fibroblasts were all transfected with increasing amounts of VO659. On the left, you can see the individual cell lines, but on the right, you can see them grouped per polyQ-length category. And what is clear here is that the normal wild-type protein, depicted by the green line, shows a milder reduction than is seen with for the mutant proteins that are the blue, yellow and orange lines. And you can see that there is a clear length dependency. So the longer the repeat, the stronger the effect. We over the course of a number of years, we've performed some studies in 4 different polyQ disease mouse models, 2 different Huntington's disease mouse models, the R62 and Q175, and also a mouse model for SCA1 and for SCA3. And what we did in all these 4 studies is we injected the oligo weekly using ICV infusions, either 6 to 8 weekly ICV infusions into the right lateral ventricle. And in all cases, in all these 4 studies, we saw a significant reduction of the mutant polyQ proteins throughout the brain and the spinal cord. And yeah, a number of these brain regions are depicted here, but the reduction was a lot more widespread than I'm showing here. What we also saw is that if we followed up the Q175 mice up to 18 weeks after last infusion, that the protein, mutant protein, was still down at this time point with no signs yet of it going back up, showing that this reduction is long-lasting. And this actually also corresponded with presence of the oligo, lasting presence of the oligo in these particular brain regions. This is an example of one of the studies we did in R62, where we wanted to also look at phenotypic benefit of the treatment. Here we use the motor rater setup, which is a state-of-the-art method allowing fully automated analysis of motor performance and gait. What you can see here in the middle is an R62 mouse of 10 weeks old that is vehicle treated, and the motor deficit in this model is very evident. As you can see, the mouse can't walk very well, it's very slow, there's a bit of a tremor here, a hunched posture. And in particular, if you compare it to a wild type mouse here on the left, of the same age. You can see this one is not doing so well. On the right-hand side, you can see again an age-matched R62 mouse that has been treated with 6 weekly ICV infusions with the with VO659, and you can see that its motor performance looks a lot better, a lot more like the wild-type mouse here on the left, and that is actually what came out of this objective analysis, that there was a shift in motor performance, several aspects of motor performance towards wild type. So where does Vico stand at the moment? So at the moment we are initiating CTA and IND enabling tox studies, and our first results are looking positive. We see that VO659 is well tolerated and also shows a rapid distribution throughout the brain and the spinal cord of monkeys. We are also able to obtain brain levels that are in the efficacious range that we saw in our mouse studies without any dose-limiting side effects. So at the moment we are on track to perform a first in-human study towards the end of 2021 in one of the polyQ disorders, and most likely this will be SCA1, but we are still debating that. So on the next slide, there's a summary of what I just tried to tell you in the last 8 minutes. I think I kept to time, and I would like to leave it here and would be happy to take any questions.
Yeah, thank you very much. There's one question, which I think is a very obvious one, which comes from Thorsten Smith. And it's on how do you avoid targeting transcription factors which have a CAG-repeat as well?
Yeah, so that is indeed a question that is very often asked. The chance of off-target effects on the wild-type proteins, and it's a very relevant question. It's also a difficult one to answer because it will really depend on, for each individual, that there will be a different profile of CAG-repeat length and structure per polyQ protein. So depending on the length and whether there are interruptions present or not, you will see a different effect. We have shown that the shorter the repeat, the less of the effect, but still there may be some off-target effects there, and these will be carefully monitored in our tox studies and in our clinical trials. Okay, thank you very much. We now move into the clinical realm, and I'm going to have a talk by Robin Wolz from IXICO on the challenges and opportunities in the deployment of volumetric MRI and SCA clinical trials.
Thanks, Tom. Can you hear me?
Yep, we do.
Great.
We can see your slides as well.
Perfect.
Yep.
Hello, everyone. My name's Robin Wolz. I'm from IXICO. So as Holm said, we're not developing drugs. We're providing imaging, CRO, and data analytics services into CNS clinical trials. And today I'll talk a little bit about, quite generally, about kinds of challenges to deploy MRI in multi-center clinical trials, and then go a little bit into the work we've done with the ESME consortium on volumetric analysis in SCA3. Just my disclosures, and then that's the outline of the talk. So starting with an overview on volumetric MRI in SCA, and I don't need to go into a lot of detail here, clearly with this group. So everyone knows that there's increasing evidence that shows the sensitivity of, in particular, longitudinal volumetric MRI. There's been some work that shows the sensitivity to detect disease progression, and so it's a widely discussed biomarker. There are then additional MRI sequences that are of increasing interest: diffusion, resting state MRI, and MRS. But today I'll focus on volumetry, and as we've seen in the previous talks and throughout the conference, obviously the regions we're interested in are around the cerebellum, different cerebellar substructures, as well as the brainstem in particular. Before we go into detail, I'll just show this slide, which shows the imaging protocol developed as part of the ReadySCA consortium. So as you see, this is a really comprehensive MRI protocol ranging for around 1 hour, including structural MRI, but then also different T2-weighted sequences, and then several advanced MRI sequences from diffusion over resting state into MRS, as well as a couple of optional sequences, susceptibility weighted imaging, MTR and ASL. So there's really a lot of data being collected in those kinds of trials and increasing evidence being developed around different biomarkers. But as I said earlier today, we'll focus on basic volumetry. So what are the, generally, what are the challenges around multi-center MRI image acquisition? What are kind of considerations that should be kept in mind when developing those protocols? So as always, the key question, or the key thing to keep in mind is a trade-off between using the most modern and available sequences, most modern scanners, and the ability to deploy in larger, increasingly sized multi-center trials. Examples are as basic as the field strength. So you may want to move to 3T, and that's available now in a good majority of research sites. But to be more inclusive to sites, you may want to consider 1.5 as well. And the trade-off you then have is harmonization that is required across the different vendors and the different sequences. And that obviously extends into the more specific MRI parameters, contrast parameters that vary slightly between scanner manufacturers and models. There's questions around resolution, which I think is an interesting one that I'd like to spend a little bit more time on. So key trade-off there is, does my analysis really benefit from an increased resolution that novel acquisition might allow? So if you're doing a standard radiology read, for example, then that may not be worth the while. Whereas if you're looking at a high resolution or really fine-grained volumetric differences, as you potentially have in the cerebellar subregions, then going from 1 millimeter to 0.8 millimeter, for example, is certainly relevant. But again, we need to make sure that it can be harmonized across the different scanners in the study. Downside is potentially increased scan time. You can address that by using accelerated sequences, which get increasingly available, but they come at a cost as well, potentially decreased signal to noise ratio or increased motion artifacts. So these are all things that should be considered before finalizing the protocol. And then once the trial starts, the kinds of things that should be kept in mind are how do we make sure we actually use the qualified sequence on the qualified scanner. The more centers that are involved, the higher the risk is that there's some local deviations. There are upgrades to the qualified scanner, which might impact scanning qualities. All of this needs to be monitored as the trial evolves to guarantee best quality data. And obviously any quality artifacts need to be monitored closely, like motion, scaling, noise, and other artifacts. So the kinds of steps that help mitigate those risks is obviously starting with a systematic and rigorous qualification step where data is collected on the scanners at local sites, rollout of specific scanning sequences depending on the scanner, and then correct setup can be assured through phantom scans, healthy volunteer scans. And during scanning, it's critical, especially when in a movement disorder, to assure correct patient positioning and field of view planning, because these are the subtle effects that can then impact things like scaling. And if you have differences in scaling between your longitudinal time points, that gets to the magnitude that then affects the ability to detect atrophy. And then, but some of these things can now be corrected or assessed quite well automatically. So automated and visual quality control are critical, and I think should be done centrally in a multi-center clinical trial to make sure everything is done at the same quality. And then finally, increasingly important as studies get longer and longer, that the tracking of scanner upgrades as we go through the study, and we can make sure that any upgrade is assessed before and after the upgrade to see whether it has any impact on the scanning quality. So that's just a very basic introduction to the operational side of running MRI in clinical trials. And I'll now spend a couple of my remaining minutes on some work we've done with the SB consortium on brain volumetry in SCA3. So at IXICO, we have a volumetric analysis solution called LEAP that uses a concept of multi-atlas image registration. So the idea is to leverage a reference database of images that represents the population, so SCA3 population or any other neurodegenerative disorder, and that can be trained to segment new images or unseen images, both cross-section and longitudinally. It applies to a large variety of different brain structures, and it can be customized to specific trials as needed. And what we have done with the SME consortium is to integrate a cerebellum sub-segmentation provided by Dietrichson and co-workers, where the cerebellum is divided into around 20 different subregions. And so the LEAP method was trained to integrate this protocol, and so it can be rolled out to new trials. And that was applied to the data retrospectively collected in the SME cohort. So I just highlighted the importance of a standardized collection. So as you see here, even with retrospectively collected data from various number of sites, you get interesting results and good statistical power, because what we see in this data set, so this is around 300 different subjects from 6 centers, and the SME consortium have obviously published on these results in more detail. And I think Jennifer Faber will be talking to some of those later in the conference as well. But just very briefly, what we see here is both by looking at the brainstem structures as well as some of the cerebellar substructures, we see good discrimination between the control subjects in the study, as well as the pre-symptomatic mutation carriers and symptomatic mutation carriers, really showing the ability of structural MRI and automated analysis to detect those changes both before clinical symptoms manifest and then obviously also differentiate further as the disease progresses. And that brings me to summary. So obviously MRI has been discussed, or neurodegeneration is widely understood in different spinocerebellar ataxia. Volumetric MRI can now be robustly rolled out in large multi-center clinical trials across all the scanners, which maybe is one of the differentiators from some of the more advanced sequences where you may have to do sub-studies and so on. So if we do volumetric T1 weighted, that's really stable now, and can be improved further with careful harmonization and controlled acquisition. And that then allows the collection of a biomarker that allows to detect changes in the pre-symptomatic stage as well as during disease progression in SCA. And that was my last slide, and I'm happy to take your questions.
Thank you very much. I think this also provided a very nice link to the working group presentation yesterday by Gylinos. As I don't see any immediate question, we leave the question for the coffee break and move on to the next presentation, which is actually on trial results. So Christopher Kenney from Cadent is going to present the trial results from Cadent.
Hi, thanks. Can you hear me okay?
Yep.
I want to start by thanking the SCA and ARCA Global leadership for this opportunity to present. For those of you who don't know me, Chris Kenney is my name. I am a full-time employee at Cadent, so that's my most important disclosure. I am a neurologist and movement disorder specialist. I trained with Joseph Jankovic when my hair was a slightly different color. So briefly on what Cadent is up to, we are modifying ion channels in hopes of modulating neurologic and psychiatric disorders. So we have an NMDA PAM in development for schizophrenia and an NMDA NAM in development for treatment-resistant depression. What we're here to talk about today is CAD-1883, which is an SK2 positive allosteric modulator that we're developing for spinocerebellar ataxia. And just a quick comment, you know, it's very exciting to sort of see all the genetic opportunities that are coming. I think in our lifetime, we're going to see some really exciting changes in these disorders and others. I think if you consider the number of genotypes for SCA and for ARCA, and then you consider ataxia more broadly in terms of the other causes like stroke and multiple sclerosis and a long list of others, I do think that it's important that we attack this both in terms of disease modification and as developing symptomatic therapies. And so that's the direction that we're heading as Cadent, is developing a symptomatic therapy for spinocerebellar ataxia. So before I get into the drug that we're developing, just a little bit on the target. So we're targeting SK2. For those of you who aren't aware of this ion channel, this is a small conductance calcium activated potassium channel. And it's incredibly important in terms of regulating the firing of Purkinje neurons in the cerebellar cortex. And so if you take a look at the Purkinje neurons in the setting of tremor, the Purkinje cells are firing in a hypersynchronous pattern. And in the setting of ataxia, there's a chaotic, sort of more irregular firing of Purkinje cells. And if you introduce an SK2 positive allosteric modulator, it does two things. One, it decreases the hypersynchronous firing and tremor. And two, it increases the regularity of the irregular firing in ataxia. And so that's our interest in the SK2 channel. The drug that we're using to target SK2 is called CAD-1883. This is a potent positive allosteric modulator of SK2. We know that we have good oral bioavailability and that we're getting exposure in the brain, not only in animals, but in humans. And we've done non-clinical experiments to show that the drug can improve ataxia and tremor. We've moved forward into the clinic and completed a phase 1 program in over 100 subjects, and the compound has been well tolerated up to 1,200 milligrams per day. The next step that we took was to complete a proof of concept in essential tremor. That study is completed. I'll share a little bit of that data in a moment. And the reason why that's so relevant is it's the same target for SCA as it is for essential tremor. And so we've sort of de-risked the program somewhat from that perspective as we transition into spinocerebellar ataxia. So we were days away from consenting our first subject in a spinocerebellar ataxia trial when COVID-19 made its way to North America. And so we put that trial on pause. And our intention, obviously there are a number of obstacles, but our intention is to restart the trial in the first half of next year. So here are the building blocks that got us to where we are. So first of all, starting all the way to the left, in addition to putting together the typical toxicology package, our Chief Scientific Officer, Tim Pizer, conducted a few different experiments, one of which was looking at cerebellar firing in a SCA2 mouse model. Another was looking at coordination in an episodic ataxia type 2 mouse model. And in both cases, we're able to improve either the Purkinje cell firing pattern or the incoordination. And what's not shown is we also completed a harmaline-induced tremor model that showed improvements there as well. I talked to you a little bit about the phase 1 trial. We started at a very low dose of 10 milligrams in a single dose and then worked our way up to 600 milligrams twice per day over 14 days, and the drug's been well tolerated in those dosages. The third figure from the left gives a summary of the essential tremor trial that was done. Obviously, we're here talking about SCA, so I'm not going to spend too much time on this, but it was a positive proof of concept trial where the majority of patients, about 3/4, showed an improvement on the TETRAS PS in response to CAD-1883. And there are two things that we learned from that trial that we want to carry over into the SCA trial. One is we want to rely heavily on a central rater, looking at videotaped ratings. In the case of essential tremor, it was the Tetris PS, and in the case of SCA, it will be the SARA. And the other thing we use is the wearable technology to confirm the findings of the clinical evaluation. And so we intend to use both of those as we go forward into spinocerebellar ataxia. And then this is the last slide that I'm going to share with you. This is the Synchrony 1 trial that we intend to start next year. So once we get it up and running again, it will be a double-blind placebo-controlled trial of 12 weeks duration. We are focused on sites in the United States with an intention to expand, obviously, global as we transition into phase 3. We're allowing multiple genotypes into the trial, with the idea being that we're hopefully developing a symptomatic therapy irrespective of genotype. So we'll be letting in SCA 1, 2, 3, 6, 7, 8, 10, 17, and also ARCA 1. And we'll be taking a look at safety, tolerability, PK. And I already mentioned the weight that we're giving to central raters. So central rater and the local rater will review the SARA. The central rater, it will be obviously a videotape SARA, and we'll also be looking at the FARS ADL for activities of daily living. If you take a look at the picture in the middle, we're going to be using wearable technology from Biosensics. So what you're seeing is a picture of the in-clinic assessment that we're focused on. There will also be a pendant that's used in the outpatient assessment that was designed by Biosensics to assess falls and other things. We're really excited about using this technology in the phase 2 trial based upon the data that we have from Insight 1, which is a non-interventional trial. Special thanks to Dr. Jeremy Schmahmann and his team at Mass General for conducting this trial. What we did is we took a look at 18 subjects, 14 with SCA and 4 controls, and found that there was really good correlation between the wearable technology, the SARA, and the bars for that matter. And also, we were able to pick up on some subtle differences between controls and SCA, which are difficult to evaluate quantitatively by a clinician, in particular turn duration and sway velocity, which are predictors of falls. So we're really excited about going forward with CAD-1883 in hopes of evaluating it in SCA as a symptomatic therapy for ataxia. And our intention is to get this trial up and running in the beginning of next year. So that's all I have. I want to thank you for this opportunity. Have a good day.
Okay, thank you very much. So I've got a question. So there's actually 2 questions. One I think is a rather funny one, which is would you consider running your phase 2 trial in New Zealand, which is COVID-free? But I was actually wondering if you have if you have discussed your using the variables for measuring ataxia with the regulators and what the results were of this discussion.
Well, so we're in relatively early development. So did we mention that we're going to do that? Absolutely. But, you know, as you're transitioning from sort of phase 1 into phase 2, it's more of the focus is really on sort of safety and less so sort of on efficacy outcomes. So I think that they are aware of what we're doing, but I would not say that there was a robust discussion back and forth. I would see that happening, you know, really at the end of phase 2.
Okay, thank you very much. Then we move to the last presentation in the industry panel, which is now really not on trial planning, but on trial results. And quite happy to have Melissa Beiner from Biohaven presenting their results. So Melissa, please go ahead. I don't hear you yet.
Okay, can you hear me now?
Yes.
Okay. I didn't realize I had to unmute that. So hi, and thank you very much for having us at this. This is a really phenomenal global conference. I think that this field is just booming. Even in just the last 3 years, it's really incredible to hear all of these presentations over yesterday and today. For those of you who don't know me, my name is Melissa Beiner, and I run the ataxia clinical trials for Biohaven Pharmaceuticals. So what I'm going to talk a little bit about today is a trial that I think most people are very familiar with. The first part of what I'm going to talk about is a little redundant. So at the last SCA Global Conference, we talked a little bit about some 48-week data we had from the open label phase of our first clinical trial with our drug troriluzole, which is a glutamate modulating agent. It's actually a prodrug of the drug riluzole. Also has some selective potassium channel activation as well. And the reason I'm giving background here, and I'm going to go through these slides very quickly, is just for sort of people on the call who aren't familiar with this study to have just a little bit of context to put this in. So we had an original, this is just, again, this slide I stuck in here to give us a little background on what we have from our natural history data across the globe to tell us about progression rates in SCAs using the SARA, which again, we've tossed around, and there's been a lot of talk throughout this conference also on outcome measures. So originally when this trial was started over 3 years ago now, right, we went with the SARA, which is actually what has been used and validated over time to study patients clinically by neurologists. So the recent meta-analysis they did that was done by Diallo on kind of natural history studies across the globe put sort of an annual progression rate of patients who are untreated with SCAs at anywhere between really about roughly 1 point to 2 point progression per year, depending on the SCA, with the more rapid progressors being closer to 2 points per year, the less rapid progressors being about 1 point per year. So I just put this in here to give a little context to what we're going to talk about with our open label study. So as I said, our trial started about 3.5 years ago, and it started as a randomized double-blind placebo-controlled trial of troriluzole. And our endpoint was at 8 weeks. And so we really didn't have a lot to go on with what we were going to see with this drug, other than a few trials that had happened in Europe with riluzole. And we did not hit our endpoint at the end of 8 weeks. So we did already have within our trial a 48-week open label phase built in. And the overwhelming majority of our patients, about 85%, ended up going into that open label and sticking out the full 48 weeks within the trial. And when we got to the end of that, we really decided to dig into the data, and it was mostly because of what we were hearing from our investigators. So over the course of that study, we had 21 trials across the United States, and we had a number of our investigators saying, look, we have patients who really feel a benefit on this drug. There's even a subset who are actually showing improvement over a year. And so we said, okay, well, what can we do at the end of this year to look at it? Because these are really just patients who are on open label drug. So what we did was, courtesy of all of our investigators who participated in the natural history studies in the US, Dr. Tia Shizawa was generous enough to lend us his data from the US longitudinal cohort. And what we did on that, so this kind of ties in what Ralph was talking about earlier in his presentation using natural history controls. We wanted to say, well, what could we tell from this data? Obviously, it's not ideal. We didn't have a placebo control arm in that open label. What could we do here? So we took patients from that natural history cohort and matched them as best as we could to patients within our trial. So we matched them on genotype, age at baseline, gender, SARA score, and severity, so initial score on their gate item of the SARA. And this just gives a little bit of what those two groups look like. The troriluzole group on the left, natural history on the right, sort of roughly matched demographically. And then with SCA's, biggest difference is really there was a much larger percentage of SCA3s in the natural history cohort. And again, this is data that a lot of people on this call are familiar with. We can, if there are any questions on it, I can stop at any point. But what I really wanted to show is just at the end of this 48-week period, what we saw here, we did actually also impose, a fixed effects ANCOVA model on this that kind of adjusted for these variables. And what we saw was patients here on the right in the red were patients who were actually on troriluzole for 1 year in the study. And what we saw was maybe a modest improvement in the SARA, but overall, what we saw was a lack of disease progression as compared to the US cohort, which had what you would expect, a little over a point progression in their SARA over the course of that year. So what do you really make of this at the end of 48 weeks? This was combined with the fact that now at this point, we had our patients were off drug. So we had investigators calling us saying, look, as patients are coming off drug, in especially a certain subset of these patients, we're hearing complaints that they're worsening. Their symptoms are coming back. They feel more unstable with their gait. Is there any way that we could get compassionate use? So, you know, initially it started with, can we get compassionate use for 10 patients? Then it grew to, you know, enough investigators saying, can we get compassionate use for 20? Because that's such a lengthy process, the easiest thing we had to do there was to extend the open label. So we added another 96 weeks of open label for these patients and decided to look at that over time. So the trial design then progresses now to, we're done with this 48 weeks of open label, and we're now instituting a 96-week extension phase. The one thing we added here was for patients who, you know, felt that they weren't dramatically improving once they were on drug, or for patients who had some loss in function, we did allow some patients to go up to a higher dose of troriluzole. And, you know, this was based on some analysis that we did from the first part of our randomization phase, showing that patients who were thinner, actually who had a higher exposure to drug, did better on the drug. So we didn't know that we were hitting our target range here with the 140. So what did we see? Over time, what we saw, the middle of this is very difficult to interpret because we ended here at open label, and then patients going into now this, what we called an expanded extension phase. A lot of those patients had a large gap in dosing off drug. That gap in dosing was anywhere from 12 weeks to patients who were off drug all the way up to a solid year. But where were we at 96 weeks? So if we look at 96 weeks overall now, we've got patients who are 96 weeks on open label, but 3 years off drug for some of those patients. And our SARA score had progressed 0.37 points overall in these patients. So to look at this graphically, these are all comers. So this is, you know, 66 patients that actually went through this whole part of the expanded extension phase in the trial. So here, can you guys see my arrow, by the way? You can see this arrow? Okay, great. So this line here running across the graph, this line is reflective of patients' SARA score when they were initially randomized into this trial. This graph begins at the end of the 48-week open label extension phase. So if you looked at all of our patients at the end of that 48 weeks, I think I showed you the 0.34 earlier. That was the minus 0.34 earlier. That was from the patients who are matched on that intersection analysis to the US longitudinal cohort. Patients here were all comers, had actually roughly a 0.85 point improvement in their SARA score at the end of that 48-week open label extension phase. This is anywhere from 3 weeks to 12 months off of troriluzole. So as you can see, there's over a point decline overall in the SARA score here. This line here reflects when patients then officially entered this expanded extension phase and went back on drug. And as you can see, their SARA score tends to stabilize over time again once they're back on drug. So we did want to look a little more closely at this gap analysis and say, okay, what does it look like for patients who were off drug for a shorter time period versus longer? And we roughly broke that down into patients who were off drug for less than 180 days. So as you can see, not as much of a dramatic decline, bit of a decline in the SARA, and this represents the time course for the next 48 weeks when they go back on drug. Patients off drug for greater than 180 days, As you can see, the decline is much more steady. And once again, back on troriluzole, overall, they stabilize out over time. And we did take this one step further. And again, anecdotal reports from some of our investigators who felt that patients who had kind of started drug earlier in the course of their disease, and also patients who represented more rapid progressors, so SCA1s, may have had the greatest benefit from this drug. And we recently, as just a little side note, because we're doing a whole sub-analysis on this now, spoke at length with a patient at Dr. Sue Perlman's site, who actually began this study over 3 years ago, SCA1 patient, young, relatively early in the course of her disease. So when she was initially randomized into the study, she was having difficulty walking up and down the stairs without some assistance, difficulty in the grocery store, was not able to wear high heels, and was having some speech issues. Over the course of the first 4 or 5 months of the study, she went back to wearing high heels, required no assistance up and down the stairs, was going to the grocery store completely on her own, and her husband noticed an improvement in her speech. That patient is now over 3 years on drug. She is still wearing high heels. She is still walking up and down the stairs. And again, this represents one patient, but it's these patients that had us kind of go through and review all this data in slicing it every possible way. So when we looked just at, so we do have some really small analyses, but the numbers are very tiny, looking at patients earlier in the course of their disease, looking at patients with more rapidly progressive disease. This just shows you what it looks like for just our SCA1s. So as you can see, you know, they really do, over the course of this study, tend to have a more dramatic decline off drug, and they really do stay the course here. So when you think about a SCA1 patient who at this point right here, here's where they were randomized on their SARA score. Here's, you know, about 0.4 points on their SARA. What you would expect really for a SCA1 patient, they're the more rapid progressors, a roughly 1.8 point progression per year. And what we're seeing is relatively no progression out to a 3-year mark. So again, overall at, you know, roughly up to 3 years, we're seeing patients with relatively little progression. So, you know, we can assume, we're assuming here that this is suggesting really an attenuation of disease progression from what you would expect with patients who are untreated based on cumulative natural history data. The other thing that was interesting is what we saw across the board was when patients experience a gap in treatment, and you can actually look at this. We've looked at all, you know, 74 patients out in individual graphs, looking at their gaps in dosing, and we saw a decline, which attenuated again once drug was resumed. So where do we stand now with this? So with this study, our patients are, we had a very official 96-week database lock where we analyzed this data. Patients still remain in this study, and right now we have our study 206 going, which is a randomized double-blind placebo-controlled trial using troriluzole over the course of 48 weeks. So this will sort of be the true test of it to see what do we see at 48 weeks with a placebo-controlled arm to this. So we're excited to see, and we're hoping to complete the study. We were hoping to complete it a little earlier. I have to say COVID got in the way of that. So we're still in a holding process. But anyway, thank you for your time. And if there are any questions, I can take those now.
Thank you very much. I think there's one question which I would like to ask you to reply to quite quickly. So the question was whether the composition of the cohort we recruited in terms of SCA types was intentional, so that you recruited quite a lot of SCA1 and 2 patients and not that many SCA3 patients, if I recall correctly.
Yeah, so originally, I mean, I think that ideally in the 201 study, we recruited 1, 2, 3, 6, 7, 8, and 10, sort of the same as for the 206 study. When you saw that original breakdown of just the 1, 2, 3, and 6, that was really because we were matching that cohort to the US study that had just looked at the natural history data for 1, 2, 3, and 6. But we did want to limit the number of SCA6s because, again, they're pretty slowly progressive, and it's going to be harder to pick up, especially when you're looking at attenuation of disease progression versus a dramatic improvement in symptoms over the course of a year. That's going to be a tougher thing to see. I think that having looked actually at all the data from 201, if we could have designed the most ideal situation for 206, we would have looked selectively at SCA1 patients early in the course of their disease. But because it's so rare, that would have been an impossible study to recruit for. So we did try to create the bulk of our patients with SCA1s, 2s, and 3s. And again, I'm sorry, go ahead.
No, just wanted to thank you for answering the question.
Okay.
Okay, thanks a lot. So actually we had planned to now move into a 30 minutes coffee break. However, as we also had planned to have a 20 minutes discussion on trial feasibility and have used up the time which was planned for this discussion, I would at least like to give Thomas and Mathis the opportunity to perhaps give a short statement on trial feasibility before we move into the coffee break and discuss the questions which were not responded by during this session. So Thomas and Mathis, if you would like, please.
Mathis, would you like to start?
You can have first go.
Okay, I think it's a pleasure to see that there are so many companies now in the field are developing drugs for spinocerebellar ataxias. My impression is that the receptors still lag a bit behind, but I'm quite confident that they may be discovered by the industry now also, and we will see developments there in the next years. I think with the one topic that appears to be very important is the question of trial design. I must say that over the years, and I've been attending ataxia conferences now since 1990, we have I've heard numerous reports of open label trials with positive, very promising results, and nothing of that was confirmed in any controlled trial. So I'm very strong in my statement. We really need placebo control. I must say that I'm also quite critical with the natural history controls that Professor Hilgers introduced, I think, yesterday in Massimo Pandolfo's presentation. We saw a striking example that placebo group was identical to the variant group in the MOXIE trial for at least 6 months, even 12 months. And if one had taken the natural history data as a control, there would have been an apparent but not true effect of the compound, which was due then to the placebo effect. So my view of that is we need this exploratory trials, the small trials with biomarkers and exploratory markers, but then the final proof must be a placebo-controlled trials, and there's no change to that. One problem that we are now facing is how we balance between the natural history studies and the various treatment trials that will come. In ReadySCA, I already see that patients leave the natural history study and enter treatment trials. So this makes these natural history studies and the results questionable. And we will also see then competition for patients, competitions by different trials for the patient. So this will be a problem in the future, in particular in the SCA3 field, because quite a number of companies now focus on SCA3, which is understandable because it's the most frequent type. So one possible solution is, and there are solutions, these are considered in US and also in Europe, that there are pre-competitive consortia of industry companies collaborating with academic centers, so that a larger number of companies could have access to natural history data, which are needed to design the interventional trials, and then that the competition begins then really with the placebo-controlled trials.
Okay, thank you. Mattis, would you like to add on that?
I don't have a statement. I just have a question, which is a twofold question. The question is asking the industry colleagues, do you think the natural history data which we have so far, do they suffice planning for your respective compound or gene therapy? And I would like to, maybe one of you saying, what is the main argument for you why the natural history data which are out there are good to go? And maybe one of you saying, no, it's not. Just with one sentence, what is the major limitation of the natural history data for the repeat scars out there? So earlier that for the HD, the wrong outcomes or not heard the outcomes she would have and correctly or not in the fashion she would this for the repeat scars. And maybe one of you saying, yes, do you think they suffice? The other one saying, not. Sentence statement.
So anyone want to comment?
Yeah, this is Chris. I don't mind commenting, although I don't know if it was just my computer, but I only caught about 60% of the words you said. So I think it's sort of an IQ test. I'll see how I do. I think our situation is, you know, a little bit different. We're thinking about a symptomatic therapy for multiple genotypes, right? And so we're going to end up with a mixture of the more rapidly progressing genotypes that Melissa mentioned, like SCA1 and 2, and also less, more slowly progressive like SCA6. And so for us, you know, we really need to have, I think, some sort of a control above a natural history. I mean, I think that, you know, my personal opinion for the others is that it has a reasonable place. I think maybe just less so for us.
And maybe I would just sort of add, I mean, so first of all, I want to say I totally agree with Thomas about the importance of a controlled clinical trial, even if it's a rare disease. I know that there are some exceptions of this, but I'm at least for as much as it is possible, a proponent of having some controlled clinical trial data to make a conclusion. So I think for us, a similar reason in spinocerebellar ataxias or other ataxias applies as for HD, that giving a therapeutic that targets particular aspects in the brain pathology, CSF samples are very important because this will really help us as we design the clinical trial. And I think the clinical trial or the clinical studies, the natural history studies in SCARs, I think it's a great effort. So I do think that they provide a good source of information, but I don't think that they are, depending on the exact nature of the trial and the exact study population, it will all depend on having a cohort that really is as closely matching the clinical trial cohort. And I think this is very difficult to say in advance before you have some sense of what the clinical trial will ultimately be.
Thank you.
Yeah, I have to say, I completely agree. And I do think that also, you know, keep in mind, the regulatory agencies are not very accepting of natural history comparisons. And which is why, right, we're doing a whole year-long trial right now in 206 with a placebo-controlled arm, having patients commit to placebo for 48 weeks. You know, that can become more difficult over time as there are competing trials. But by the same token, I think to just give you some clinical information to base trials off of, they're invaluable, right? So while you can't look at this data 3 years out and draw any definitive conclusions, and we certainly wouldn't be able to take that to the FDA to say, okay, we want a label, I do think it gave us a good leg to stand in and saying it's really worth testing this drug for a year, because to have patients, especially with SCA1, who have no progression in their SARA at 3 years, it would fly in the face of what we see in the natural history data, which is a definitive progression over 3 years, especially in this patient population. So it argues strongly to be able to use that data to say, look, you know, there's definitely a signal.